Hello everybody and welcome to the third webinar of the series. Today I'm going to be talking about supplementation. So my name is Emily Perry and I'm interested in. So as for the previous sessions we have muted the mic, so please let us know in the Slack work session. Astrid and Erin are here with me, ready to answer all your questions, and if I need to be stopped and interrupted, they will do so. So, as with the previous sessions, they're available on the Train Online course. And as with today's session, you can get help with the exercises on the Slack workspace, and you can email us as well if you don't want to use Slack. So, I'm going to talk about Ensembl Regulation, which is the first part of this third webinar. And Erin will be following it up today with data export with biomarkers. As with the previous sessions, I'm going to talk about how we annotate regulatory features. I'm going to give a demo of how you can get the data, and then the exercises will be available for you online. So what we're going to talk about is epigenetics in gene regulation. We're going to talk about some of the methods we use to study epigenetics. And then I'm going to talk about what we do in Ensembl Regulation, where we get the data from, and how we process that to produce our regulatory build. So epigenetics is the study of heritable genetic changes without any changes in the DNA sequence. And it is known that this regulates gene expression. An epigenetic change is very important for cell differentiation. So this is just a schematic showing cell differentiation of the hematopoietic stem cell down into the many, many other different cell types that it leads to. And you can have cells which have come from the same source, they're genetically identical, and yet they carry out very different functions, they are morphologically very different, they express different genes, and this is down to the fact that they have different epigenomes. And one of the sort of classic diagrams of how epigenetic cell differentiation works is that we have this marble run with the stem cell at the top of the marble run, and as it rolls down the marble run, it can go one way or it can go another way, but it can't go, crucially, back up. So the methods of gene regulation, epigenetic gene regulation we're going to talk about today include histone modifications, which we study via ChIP-seq, transcription factor binding, which we also study with ChIP-seq, and open or closed chromatin. And we're talking about data from DNase sensitivity, although there are more modern methods of studying open chromatin these days. So this all comes down to this model of how promoters and enhancers work. So this line across the middle of the screen is represents the DNA, and it has a gene. And the gene has a region at the 5' end, which is known as the promoter. And the purpose of the promoter is for DNA polymerase to bind. But DNA polymerase doesn't just go around binding DNA willy-nilly. It needs help. It needs help from transcription factors. So these are a number of different proteins which, in response to different stimuli, will bind to the promoter, and they will bring DNA polymerase in. But it's not completely that simple, because sometimes these transcription factors bound to the promoter can't do it by themselves. They actually need an enhancer, which will be a region of DNA elsewhere, usually on the same chromosome, and that binds further transcription factors, and it's believed that these regions are brought physically into contact with each other, which then allows DNA polymerase to be bound and transcription to occur. So transcription factor binding at promoters and enhancers is necessary for transcription, and this is the combination of epigenetic marks which affect the ability and the probability of transcription factor binding to occur. Transcription factor motifs are short sequences that attract transcription factors, and they are scored out of 1 for strength, where 1 is the strongest and 0 is the weakest. And we currently use transcription factor motifs from Jasper. Another major part of what we're looking at is histone modifications. So histones are these very large proteins which are made up of of 8 subunits. So there's 2 H2As, 2 H2Bs, 2 H3s, and 2 H4, and they form this massive blobby protein, which then has DNA wrapped around it twice, and this forms the beads on the string structure of DNA. The really cool and important thing about histones, apart from the fact that they wrap DNA up, is that they have these big dangly long tails on each of the proteins. And the dangly tails are rich in lysines and arginines, which can both be covalently modified with methyl groups and acetyl groups. And these affect transcription factor binding and affect transcription. And the convention for writing them is just written at the bottom here. So we have this example of H3, so that's histone 3, K36, that's lysine 36, and then Me3, so that's trimethylation. So you can also have acetylation, monomethylation, dimethylation. And there's this histone code where some histone modifications are known to promote transcription and some are known to block transcription. Others don't really exist, or they're a bit dubious in their roles. And we study histone modifications and transcription factor binding using ChIP-seq. And it's important to understand how ChIP-seq works because it helps us to understand what the markers we're actually looking at are when we look at these data. So when we do ChIP-seq, we have DNA and it's bound to a DNA binding protein. The first thing you do is you carry out covalent cross-linking, which attaches the DNA binding protein to the DNA. It can no longer be washed off. You then shear the genome, and this uses a random method. Things like sonication or digestion with enzymes are the two ways that people usually go about this. So we get random chunks of DNA, which we can then use a protein, an antibody to pull down a protein. So we've got our DNA binding protein, we use an antibody to that protein, and we end up with just the bits of DNA which are bound to that protein. We can remove the cross-links, wash, sequence the fragments, and then we map them onto the genome and identify where that histone modification is occurring or where that transcription factor is binding. Now, one of the things to remember about this is because of the method of ChIP-seq, we actually get quite fuzzy edges on ChIP-seq peaks. So if you look at a transcription factor binding motif, so this example given here is actually a very large motif. It's a ChIP-seq and it's 19 bases long. Many transcription factor binding motifs are only 8 or 6 bases long, so they're really very small. The first thing to note is that when we do the fragmentation of the genome, we get very large fragments. So sonication results in fragments 200 bases long. So even though we've only got these 19 bases that are attracting this transcription factor, we get fragments which are 200 bases. We then carry out next generation sequencing, getting reads of about 50 bases. This results in something that looks a bit like this in terms of our analysis. Our motif is very small in the middle, but as much as 200 bases in either direction, because the cutting is random, means that we get this kind of peak across our motif, rather than just getting binding at the position of the motif. Another thing we look at is open and closed chromatin. So here I've got a simplified diagram of DNA bound to histones. And you'll see that some histones are bunched up really close together. Other histones are really spaced out. And we call these regions where they're bunched up closed chromatin, and those regions where they're spaced out open chromatin. And we find that transcription occurs in regions where there is open chromatin. Open chromatin is transcriptionally active and closed chromatin is inactive. And we can measure where we have open and closed chromatin with a number of methods, and one of the ones that we use and that we have data from is DNase sensitivity. So this uses an enzyme DNase to cut up the DNA, and then we purify the fragments, which we then sequence and compare the reference to identify where we actually get binding. And so we get all these fragments in what are known as DNase hypersensitive sites. So Ensembl Regulation is working with all these different kinds of data. The aim is to annotate the genome with features that may play a role in the transcriptional regulation of genes. So we look at predicted open and closed chromatin, we look at transcription factor binding, we look at epigenetic marks such as the histone modifications, and we also look at RNA polymerase binding as well, which is obviously involved in transcription. And we have data from a number of sources. So we have data for human, we have data from ENCODE, from Roadmap Epigenomics, and Blueprint for human, and we have data from ENCODE from mouse as well. So for human, we have 48 cultured cell lines from Roadmap and ENCODE, and 20 primary cell lines from the hematopoietic cell lineage from Blueprint. And in mouse, we have 79 cultured cell lines. And we have loads and loads of data from these. We have histone modifications, transcription factor binding site, DNase sensitivity. So what do we do with these data? Well, firstly, we only show a subset of the cell types. So there are more than 48 cultured cell lines in ENCODE and Roadmap Epigenomics. But we do this analysis on which we call our regulatory build. And to do that, we require ChIP-seq binding, DNase sensitivity, H3K4 trimethylation, H3K27 trimethylation, and H3K36 trimethylation as a minimum. If a cell type has all of those bits of data, we will do our analysis on them. If a cell type does not have that, we will not do our analysis. So what we do is we take these raw data from all these different sources, and we use it to predict the position of features such as promoters, enhancers, and insulators. And we tell you what the activity is in those different cell types. So this is our start point. Here we have a genomic region, and we have binding of transcription factors and histone modifications. And you'll see that we have the actual signal, the number of reads that we have of each of these different ChIP-seq experiments, and we have the peaks showing where we have the highest number of reads, the regions which we think are significant. And we find that these tend to come in clusters. We will find regions where we get a number of different transcription factors, different histone modifications occurring together. And where we get these clusters, we look across the genome to see if we can find other clusters. And it turns out that there will be similar clusters, not always identical, but similar clusters of the same sets of transcription factors and histone modifications across the whole genome. Now, if we know the function of one or two of these, that means that we can extrapolate the function of all of them. We can say, well, actually, they must all be promoters. If this is a promoter, then this thing that looks the same as it must also be a promoter. If it looks like a duck and quacks like a duck and walks like a duck, it's probably a duck, as they say. But we don't find the same promoter at the same pattern at every promoter. We may find another pattern elsewhere in the genome, which also has known to have promoter function, and another different pattern which is known to have promoter function. So actually, there are loads and loads of these different patterns across the genome. We can't just say, if you've got this, 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 and this, it's a promoter, because actually, that's not always the case. There are loads and loads of different ways that you can make a promoter. So we use this data to produce what we call segmentation. So in each cell type, we will calculate what we believe each part of the genome is doing. One of the cool things about this is it's a completely blind algorithm. It has no idea what the patterns it's looking at are. So we don't tell this algorithm whether histone modifications are activating or repressing. So what we can do then is we can use this as a test. If we've said that, we'll just find things that look like other things, and then all of the things that it finds that say are activating actually have activating modifications, that proves that it's worked, that it's done what we intended it to do. So what we do then is we have this data, this segmentation across all these different cell types, and we line them up. We put all of the different cell types together and we say, okay, what function do we have at each genomic region? And we actually do this using 200 base pair bins. And we move across the genome 200 bases at a time, and we say, in this particular region, what's happening across all the different cell types. And we can say, well, if there's promoter activity in one cell type, then this region must have promoter activity. And so we move across to the next 200 bases. Again, does it have promoter activity or any other kind of activity? And we're moving across the whole thing 200 bases at a time, identifying where we have certain kinds of functionality. And then we compare it back to the cells and we say, okay, we've said that this region is a promoter, this region is an enhancer, and it's active in this cell type, it's inactive in this cell type. And this is what you'll actually see when you look at the regulatory build. The promoters that we've predicted, or enhancers or whatever, and whether or not they're active in different cell types. So there are loads of different kinds of features that we annotate: transcription start sites, promoters, enhancers, CTCF binding sites, and what we call unannotated transcription factor binding sites, which is where we have transcription factor binding, but not really anything else. We can't really tell anything more about it. And this covers around 10% of the genome has got some kind of activity going on that regulates genes. What we don't do, we don't link these to genes. Promoters, you can reasonably infer what gene they belong to, but enhancers, insulators, other regulatory features, we don't actually know what genes they're linked to. And this is something that is being worked on in a long-term sense through things like EQTLs and high-C type data. But we don't currently have this data at a large enough scale to be able to make these links. We also don't link regulatory feature to gene expression. There is cell type specific regulation data, there is tissue specific expression data, and again, we're letting you make your own inferences, link this together. This data is very, very, very complex, and it is very new. There isn't a kind of general consensus on how this should all be done, or comprehensive database. One thing that people always ask is, well, is this a real promoter? Is this a real enhancer? What I can say is that our predictions are based on real biological data. This is not just sequence analysis. This is actually looking at binding of epigenetic markers and transcription factors. We have evidence to suggest they're doing what they think we are. We have done analyses on this, things like the blind segmentation analysis. But most of them have not been experimentally validated. They haven't been cloned alongside a gene and tested. So more data will refine and improve the pipeline. Another thing we'll do is we will predict the effects of variants on regulatory features. So the VET that you saw in the last webinar, one of the things you can get out is regulatory features and also transcription factor binding motifs. There is loads more information in a couple of papers looking at the first paper. The regulatory build looks at how we actually did this in quite a bit of detail. You can see how the algorithms were run. And the second paper, regulation resources, looks more at how you can get access to these data. So we're going to have a look at the region of the gene called LIMD2. We're going to find regulatory features in the region of the gene, and we're going to explore what cell types they're active in and how we know this. So I'm popping out of my presentation, and can you all still see that? Good. And I've gone into Ensembl, and I'm going to put LIMD2 into the search box. And instead of going to the gene page, I'm going to click on the link to the region just below it. And I'm just going to drag out a box so that I can see a bit more than just the LIMD2 gene. I'm going to look at the region just around it. So here we have LIMD2 in the middle. So you can see it's a reverse stranded gene. So if I'm looking for promoters, I want to look over here, not over here. And if I scroll further down, you'll see that the regulatory build track is on by default, and there's a promoter annotated here. Now, this promoter is actually massive. It covers most of the LIMD2 gene itself. So I always forget to mention this. The reason I know it's a promoter is because I know the colours off by heart, but there is a legend down here. So we know that red is promoter, pink is promoter flank, and cyan is ChIP-seq. So you can see we've got a promoter and we've got the pink flanks either side. If I click on it as well, you can see that it says it's promoter. It gives me the stable ID, and I can click through to that. I'm just going to close it for a second, just to point out these black lines through it. These are actually transcription factor binding motifs. And they're currently broken, but they should at some point in future, again, if you click on them, it will tell you what they're motifs for. So I'm going to click on the stable ID here, which will take me to the regulation tab. So this has all the data we want to see about our regulatory feature. So here we can see a graphic with it in context. And if I scroll further down the page, it lists all the cell types which have been studied, where it's active, inactive, and poised. So poised just means it's not active, but it's got most of what it needs to become active, and it'll just take one little thing to trigger it, is the best way I can think of to explain it. The analogy that works best is active is Usain Bolt running, inactive is Usain Bolt sitting on a chair, and poised is Usain Bolt in the blocks, ready to go. It's ready, it's just not doing it quite yet. So we have a list of the cell types where there's activity and inactivity. It's currently just not showing any particular cell types, it's just showing it the context, but I could select cells and pick a couple of cell types, or I could do all on and see whether it's active or not in a graphical way. And in both these cases, I happen to by chance have picked two where it's poised and it shows up as purple. This is quite nice, this ChIP-seq binding site here. You can see that it's inactive in fetal adrenal gland because it's got this candy stripe pattern, and it's actually repressed, which is Usain Bolt tied to a chair, in grey, this particular one in the HuVec cell line. I'm going to go into details by cell type because that allows me to see actually the data that's used to make these predictions. So if I go in here, you'll see I've got the HuVec cell turned on and visible, but I can show more cells as well. So I'm just going to go into select cells, and I'm going to pick healers and HepG2. I think these will give us a nice contrast of different kinds of activities. So yeah, oh no, HepG2 was no good, never mind. I'll carry on anyway. Healer, you can see it's inactive, and the HepG2 is another one where it's poised. I can also select evidence. So this is showing me all of the different transcription factors and all of the histone modifications that it's possible to see. If I select all on, that selects all of those for me. And if I close that, I'm now going to see peaks, the points where we have significant binding for all of these different factors. So here in the HuVec cell, so this whole region with this kind of blue behind it is the activity in the HuVec cells where it's poised. So you can see we've got ChIP-seq binding, DNase 1. We've got 3 different histone modifications. So we've got 2 different modifications of H3K4. If I look at this healer where it's inactive, you can see, excuse me, there's actually a lot less stuff bound at this particular locus. And this is probably why it's been listed as inactive. If I go further down, you can see that we've got similar kind of thing in the poised HepG2 as we saw in the HuVec, not identical. It's worth pointing out that there isn't always the same amount of data for all the different cell types. So it could be that this NRSF is actually bound to this region in the HUVEC, but there isn't data available. So it's not always clear how much is due to the data available and how much is due to what's actually happening. So another way, if you again prefer tables to visuals, if you go into source data, you can see all of this data, every single peak listed as a big long table across this region. So if you like looking at data like that, you can do. It's going to take some time to load. Rather than wait for that, I'm just going to hop back to the location tab, because you can do similar manipulations in the location tab as you can in the regulation tab. The benefit of doing it in the regulation tab is that you can see then across multiple regulatory features that may be near to each other. So if I go into the configure this page menu from here, and I go into regulatory features, I can then see the epigenomic activity, whether these transcription factors, whether this regulatory feature is active in these different cell types. So if I pick some cells, I'm going to go for H1ESC, HUVEC, and HeLa. And if I tick this and go back down, you can see again I've got this indication of whether they're active or not in these different cell types. Another thing I can do is I can actually see the evidence. So if I go into open chromatin and transcription factor binding sites in this menu, I have this tutorial which I'm just going to hide. You can see I have all the different cell types listed on the top and all the different transcription factors listed down the side. Now, where it's this dark grey, it means the data is not available. This light grey means the data is available but hasn't been displayed. And this blue means that it has been displayed. So what I can do is I can select all by transcript. I can also select all by cell type. So I'm going to select all for my HUVEC cells again. And you'll see it's now highlighted everything's there. And it's also given me a track style for this cell type. So even though there's stuff highlighted for some of the other cell types, these won't actually display until you select a track style for that cell type. If I go into histones and polymerases just underneath, you'll see that I get a very similar thing, but now I've got histone modifications. I'm going to find my HUVEC cells. And the track styles I can pick are peaks, signal, or both. So I can get the squiggly line or the blocks. I can get both of them. If I save and close that one. In this HUVEC cell line, just as I had before, I have this data, the binding of these transcription factors, the histone modifications across this region. But I can look at a much larger region rather than just focusing on a single regulatory feature if I want to. So I'm going to show you one last thing with regulatory features. I'm just going to go back to my home page. And one of the things that you can do is you can add track hubs. So if I go into human and display your data, one of the things that you can go to is track hub registry search. Now the track hub registry is really cool. If you have data that you want to share publicly that can be plotted onto a genome, you can deposit it in the track hub registry, and you can also use it to access data from other sources. So what I can do is I can search for data that interests me. So if I search for blueprint, it actually tells me that I've got one, there is a track hub, so I can attach this track hub. So this will now display the data from blueprint in the genome browser. So if I now click on this configure this hub, doesn't seem to want to display it. So what would happen if this was actually working? I'm not quite sure why it isn't. Is actually my region in detail view, my configure menu, would give me a nice big menu which had loads more options to add data from this track hub. And you can interact with it in the same way that you can interact with data that's provided by Ensembl, which is really cool. Right, I'm going to hop back into my presentation now. That was everything I wanted to say about the regulatory build. I've seen Astrid and Erin sat typing away, so it looks like people have been asking lots of questions about this in the Slack workspace. Is there anything I need to go over, or are we all up to date on that? Okay, we are up to date, so I will carry on. As mentioned a million times already, the course exercises are available in the Train Online course, and you can get help with those, or just ask us general questions using either the Slack workspace or by emailing us. The next webinar will be Erin. The next topic will be Erin talking about data export with Biomark. So this is a tool for bulk exporting data, which allows you to select which data you want to see. So we're going to learn the basics of running a query and then explore some of the options that are available. And thank you to all the team who have worked on this and all our funders.